This is the third set of slides for cell structures. We watched the first one in class on Friday, and you should have watched part 2 during the weekend. You will also be watching this during the weekend, part 3. The cell membrane is really dynamic and very intricate. It has lots of parts. And if you remember, there was an open response question that we did just based on this, and you did not have a ton of background information on the cell membrane. Luckily, for your actual AP test, you will have a ton of background information on the cell membrane, which will allow answering a question like this to happen much more easily. It regulates materials that are transported across the membrane. We call it selectively permeable or the semi-permeable membrane, meaning some things can go across and some can't. There are lots of images or videos of this. Feel free to look it up if you would like to. So the phospholipids are the molecules that make up the membrane. All cells have this plasma membrane, and many of their organelles also have the same membrane. So if you recall from the last set of videos, the nucleus, the endoplasmic reticulum, the Golgi apparatus, all has this same membrane. It is made of a phosphate head and a lipid tail. The phosphate is obviously to the left, and the two tails are to the right. It is hydrophobic and hydrophilic, which is interesting because we know hydrophilic, water-loving, and hydrophobic, water-fearing. And so the head, which is facing outwards and to the inside of the cell, is hydrophilic, water-loving, and the hydrophobic parts are going to be on the inside of the two heads. Because if you remember, the tails face each other, and the two heads either face into the cell or out of the cell. Looks similar to this. This is how they are arranged. The membrane has 2 layers of these phospholipids, and they go tail to tail. If we look at the bottom image, the heads on the top would be facing outside of the cell. The ones on the bottom would be facing inside the cell. How do these move? Phospholipids have the ability to move laterally, but only on rare occasion are they going to make a 180 turn. So these cells can kind of switch with the cell next to it. That happens very often, over thousands of times per second, whereas they can switch with the one across from them maybe once per month. We'll talk about why. And if we look back, we'll see that when you mix cells together, they can sometimes exchange phospholipids with each other, which makes it really interesting, but not important for now. Saturated versus unsaturated phospholipids. Membranes are more fluid when they contain more unsaturated fatty acids within their phospholipids. More unsaturated fatty acids result in an increased distance between the lipids, which makes it more fluid. We look at the one to the left. It allows the tails to kink a little bit, to push the phospholipids away from each other. It makes the membrane a lot more flexible, whereas a saturated hydrocarbon tail, saturated, right, we've got a lot of them, the tails don't really kink, they don't push each other apart, so we get a very viscous or slow-moving, inflexible membrane. And when we talk about cholesterol, it is found in the cell membranes of animals but not plants, and it affects the fluidity of the membrane. So the more cholesterol, the more fluid those membranes are going to be. So keep in mind we've got our phospholipids, we've got a little bit of cholesterol in our membrane, and then we have proteins inserted into the membranes, and there are a couple types. The first is a transport protein, or we call them permeases. Transport molecules across the protein. A specific type would be an aquaporin, which moves water across the membrane. The reason we need them is because there is the hydrophobic part of the phospholipids, which doesn't allow it to naturally diffuse across the membrane. The second type is your enzymes. They perform enzymatic activity. And then we have signaling molecules, which start the signal transduction pathway, which we've kind of looked at before. there are lots of simulations of this. I would highly recommend looking one up on YouTube. Just type in plasma membrane into YouTube, or the simulation of the aquaporin, which is how water moves through those transport proteins. The function of other membrane proteins, so I talked about this a little bit. One of them is the enzymatic ones that speed up a reaction. The other is a receptor. So, for example, insulin doesn't ever go into my cells. Insulin has to do with my processing of sugar, but it will bind to a receptor on the membrane. The other things that it might do are be cell-to-cell recognition. This has a lot to do with the immune system, which deals with recognizing self versus non-self cells. And then there are intracellular joining cells, which, similar to like the attachment to cytoskeleton and stuff, these provide like attachments for cells to hang on to each other for. We call this, the kind of big picture, the fluid mosaic model. Essentially, the term fluid refers to the fact that it is very flexible and constantly moving, and the mosaic, if you've ever seen like a glass mosaic in a church, refers to all of the little bits and pieces that make up this cell membrane, right? We've got our phospholipids, we've got our cholesterol, we've got our different transport proteins. It's all there doing different things. The size of the plasma membrane is increased and decreased with the interaction of vesicles. So the actual size of my cell can get bigger or smaller. We know that if I have a vesicle, let's say filled with waste, and I want to dump that waste out of my cell, it's going to fuse with the cell membrane. It's going to open up to the outside of the cell and release that waste or that secreted protein. And the old vesicle becomes part of the cell membrane, making the cell membrane a little bit bigger for the time being. When I make a vesicle going inwards, I'm going to lose that little bit of cell membrane, decreasing the surface area. Again, this is just a really good image of what that looks like. We've got our fluid mosaic model.